Hello, welcome to NCUR 2021. My name is Zachary Emmel, and I'll be presenting my research on using surface plasmon resonance to measure cooperativity binding of DNA and analyzing the kinetics of that cooperative binding. Little about me, I am currently a senior at Montana State University, dual majoring in chemical engineering and biological engineering. And I will be graduating this semester, which is very, very exciting, and I'm just a few short weeks away, so very happy about that. I'm originally from a very small town in Colorado called Elizabeth, and I came to Montana for the great engineering program at MSU and the amazing outdoors, so it was just a great fit right away. And halfway into my college career, I was able to find and work under Dr. Stephanie McCullough in her lab as an undergraduate research assistant. So I have been working on the surface plasmon resonance project for 2 years now. This is the poster that I will be presenting on at NCUR 2021. And so I very much look forward to getting to go through all of this in finer detail with everyone. But for now, I'm just going to cover a few short introductory topics. The main topic I want to cover is what is cooperative binding. Cooperative binding is the binding that occurs when multiple species bind to a single receptor. Positive cooperative binding is when the first binding event influences and increases the affinity and thermodynamic favorability of the second binding event at that same receptor. That can be a bit wordy, so the key takeaway from positive cooperative binding is that the first binding event helps out the second binding event and makes it more likely and faster. An important note is that cooperative binding can also be negative, which is the opposite, meaning that the first binding event then kind of inhibits or slows down the second binding event. We're using surface plasmon resonance to study the kinetics of this interaction and of the cooperative binding of DNA. And we're doing that by observing the wavelength shift here. And I use this diagram from NCUR, where we initially bind our ligand on our glass and gold nanoparticle sensor chip. We shine light through it through an optical detector. Then when we inject our inner light and our analyte flows over the ligand, a natural wavelength shift will occur as that interaction takes place. And so the SPR machine from NCUR that we are using takes that wavelength shift and can output that into information and data file that we can then analyze the kinetics on. So again, this was just a very brief intro to this project. I hope that you'll stop by where I can cover a lot more about the Hill equation, our experimental protocol, kind of the steps of SPR that we're currently going through, and the plans we have for the future of our work. So I look forward to getting to talk about this at NCUR 2021. Thank you.